Hello, everybody. Thank you for coming. Thank you for joining us. My name is Dan Treiman, and I'm the CEO of NEIBC. It's called New England Israel Business Council. It's an organization, not-for-profit, that its mission is to strengthen the business relationship between Israel and New England. We have a great session today. We had a great help, a collaborator that helped us put it together. It's an organization called NOGAFEM. And I would like to invite one of the founder, Rna, to say a few words. Rna, the floor is yours.
Thank you, Dan. We're very, very excited to be here with you tonight. And thanks to NEIBC for allowing us to co-host this event. NOGAFEM is also a non-for-profit organization looking at accelerating women's health technologies. We particularly focus on the inclusion of sex and gender within women's health technology design thinking to try and match high unmet needs towards personalized solutions. We're very, very excited to bring you the high caliber Israeli innovation that we are sure is going to transform women's health. And without further ado, I hand back to Dan. Thank you and enjoy the session.
Thank you, Rna. Thank you. Another thank you, of course, to the panelists, Shlomit, Tzafrir, and Ronen, and of course to our moderator, David Ufer. And with that, I'll transfer the talking stick to David. David, the floor is yours.
Thanks, Dan. Appreciate the opportunity to be here. NEIBC has curated over 1,000 startups throughout Israel, Startup Nation. And within that, we've identified over 100 women's health startups just in Israel alone. So we've decided to put together a series of webinars with a different theme for continuing throughout the year to highlight and introduce these to the general healthcare community. In the last series, we had companies device related within pelvic health. So today's 3 companies we've chosen out of that entire landscape that are AI-driven women's health technologies. I myself have been involved in women's health for 30 years, anywhere from diagnostics, breast health, pelvic, IVD, infertility, mental health. It's quite a passion for me, so I'm very appreciative of being invited to help moderate here. I'm a senior partner at Alira Health, where we are an advisory firm and work with many women's health companies. I'm excited to present to you PredictaMed, Vertigo, and Barcode Nanotech. Without further ado, I'd like to introduce Shlomit Steinberg-Koch with PredictaMed. Shlomit will take us through, again, a 15-minute presentation. And anybody please put in the chat anything you'd like to comment on throughout the presentation.
Okay, thank you so much, David and Dan. I appreciate the opportunity to present here, PredictaMed. So PredictaMed is connecting the dots of undiagnosed autoimmune diseases, which is a huge underserved market. And I would like to start with a video that demonstrates the problem from the point of view of the patient. And with PredictaMed believes that this is the most important point of view. So we'll start with that.
My first symptom was at birth, came out with an allergic reaction. Had problems until a time, I didn't have teeth come in properly, panic attacks at 12. 15 is when I finally started saying, there's something wrong. And I'm thinking and pointing to my gut. And it was from 15 to 35 that the journey continued to try and get an answer to what was happening to me.
After 35 long years, Jennifer Esposito, an American actress, found that she was suffering all these years from an undiagnosed celiac disease. And this is the story of 50 million Americans. 78% of them are actually women that suffer from autoimmune diseases. Unfortunately, it takes these patients more than 4 years and at least 4 different doctors to get a correct diagnosis. And beside the fact that these patients are going through a very frustrating journey, also billions of dollars are spent by the healthcare system, and a lot of it can be saved by early detection of these diseases. The way we see the problem, when you go to your PCP and you tell him that you have a migraine, you would probably get a treatment to the migraine. But the chances that during these 5 minutes of a visit, he'll have time to review all your medical history and connect all the dots and find an underlying autoimmune disease is really low. And that's why they tend to miss these diseases for many, many years. So we at Predictamed have built the first decision support platform for early detection and intervention selection of autoimmune-related diseases. And our solution is based on 4 steps. We aggregate data from EMRs and claims. We enrich it and analyze it using our deep learning platform. We identify which patients have an underlying autoimmune disease, and then we alert the primary care physician and enable him to intervene much earlier. So we're not creating a standalone solution. We do not believe in that. We are working with the largest EMR systems to integrate our solutions within theirs. So eventually the physician will have a seamless workflow integration, and that's really a key for them. So here you can see a demonstration of our integration with Epic. You can see that we found a patient with high probability for celiac disease, and our solution is fully explainable. We can actually show based on which parameters we suspected that these patients had celiac, and this is also a key for the primary care physicians. I have a very talented team of experts. My background is in biomedical engineering. I also hold an MBA. My first startup was in the field of melanoma detection based on an algorithm I developed. But back in 2012, telehealth and AI in healthcare just started, and we were a bit before the market. So we had to close the company. Then I joined Mazor Robotics, a company that was in a better timing. So the company was eventually acquired by Medtronic in a huge deal, and I was working there for the past 6 years in leading positions. And I also became the first employee of the innovation department. The founder of Mazor, Professor Moshe Shoam, became the first investor at Predictamed and is working with us until today. Benny Gates, my co-founder and CTO, has been leading AI teams in the past 25 years. He's been working in 2 startups that were acquired, and also at Intel and Samsung in leading R&D positions. And he was the CTO of KHAT, a cybersecurity company. We have top leading medical advisors. Some of them are, or most of them are, world-renowned KOLs in the field of autoimmune diseases. You can see them listed here. And we have top leading investors and strategic advisors. As I mentioned, Professor Shoam, Professor Yossi Matias, who is the VP Research and Engineering at Google, Jack Friedman, who is here today. He's the former CEO of Providence Healthcare. He's been leading Providence for the past 25 years, and he's joined us. And Yuval Ziv, he's the founder and the CEO of Asia Healthcare Services, a primary care organization in Israel, and also joined us. And you are now bringing on board a female US-based international expert in health equity and primary care. And together we are working to address gaps in care. So the differentiated value proposition is that early detection eventually allows optimized and cost-effective intervention. And by that, we align the incentives of all the 4 different stakeholders: patients, payers, providers, and pharma company. All can benefit from early detection of autoimmune diseases. Our differentiation is based on 4 pillars. First of all, our technology is patent pending. We submitted few patents from day one. We use advanced learning methods, which I'll discuss a bit later. And we build our system in a way that it will behave real time, but also on demand. We have domain experts both in AI and in the autoimmune space. We have a proprietary data. We use data enrichment techniques, and we've built the system both on EMR and claims. And eventually the end customer value is that we shorten the time to get diagnosis, and we are doing it in a fully explainable way. And also we, as I mentioned, integrate into the workflow seamlessly. Our business model is divided into selling to providers and payers, both in a SaaS licensing model. And we did a simulation to see what's the potential market, and we saw that it's huge. So only in IBD, Crohn's and colitis, we can reach a cost saving of almost $2 billion. And if we look just at the 6 autoimmune diseases we started with, actually we have 7 to date, we can save more than $4 billion, which is quite significant. We've partnered with second largest Israeli HMO, Maccabi, with a US-based company, and together we raised to date more than 4 million patients' records. And we are using cutting-edge deep learning techniques, including self-supervised learning, the deep classification network, NLP, and others. You can see that we validated our solution, and we've built it for the 7 most prevalent autoimmune diseases. All of them, by the way, except for celiac, have medications, but celiac is also going to have a medication soon. And you can see the predominance of women in these diseases, which is quite significant. We have very promising results in our POC, and now we show that we can reduce the time to diagnosis by up to 4 years, which is quite huge. So to summarize the company traction, we launched a company on January 2020. We submitted several patents. We signed on design partnership with leading organizations, participated in accelerators like Mass Challenge, Microsoft, and Amazon. We raised pre-seed funding of $1.5 million, conducted a POC, including a study with a pharma company, one of the top 5 leading pharma companies. We also won Shield of Tech, which is an international females-led competition, a global one. And now we're closing our seed round, which would take us to the next milestone, which is doing pilots and partnerships in the US with leading organizations, for example, Hog, Stanford, CNH. Some of them are listed here, some of them are not. But we have a very significant traction in the US, and we're planning to launch our product to market by the end of the year. Join us. We're looking for people who would join in our mission to change the lives of millions. And we're looking to partner with healthcare organizations, providers, payers, and pharma companies. Thank you so much for listening.
Shlomo, thank you very much for very comprehensive description of the company and the technology. One of the questions that comes to mind, and I guess we could say this would be for all the presenters here, but we see a lot of companies that position themselves as AI, and I find them as truly algorithms, that there's not a lot of artificial intelligence that continues to go to greater levels of fidelity and predictive analytics based on the static nature of it. Could you tell us about your platform, and will this continue to improve itself, or you have an algorithm that has already been predefined and will remain static?
So first of all, it's very true what you're saying. Many companies mainly in healthcare that are saying that their AI solutions are mostly using rule-based methods. So using existing protocols and very simple methods of rule-based. Sometimes there is a success there, so I can't say that it's, that AI is better, but we're doing AI. I mean, we are doing machine learning. We are not doing rule-based methods. And I think that what's the strongest thing about our system is that, first of all, we combine the most cutting-edge deep learning techniques like neural networks, for example, and it allows us to look at tens and hundreds of parameters at the same time and build pattern of each disease. So it's our strengths, and also the fact that we use explainability methods, which is something quite new. And our system is not black box. It can actually explain itself to the physicians. And that's really important, because then we can say that you are a decision support solution for the physician. You are not a black box. They're saying him, hey, there's celiac, and that's it. We are showing the parameters, and this is really a key. Answering your second question about the ability of the system to learn and to evolve, that's true. We are doing that. So with each data set that we're getting, we're improving the system, and we're it's becoming more robust, because eventually AI is being fed by data. And as more data you have, the system would be stronger and more robust. And that's exactly what we are doing. So we have predefined models built on the 4 million records I mentioned, but now we are going to get new data sets from our partners in the US, and we are going to improve our models. Also, within each organization, each partnership that we are entering, we always need to readjust the model to fit the organization, mainly if it's, for example, in different state and the ethnicity of the people is different, for example, the population looks different, then the model need to be a bit readjusted to fit the population best.
Thank you. Very good point. Let's go lateral and process of company formation. The first question I would have is, you're just placing your seed capital. The first thing you need to do beyond develop your technology and your value proposition and your minimally viable product, what is your regulatory pathway?
So currently we do not need a regulatory approval because we are a decision support platform with very strong explainability capabilities, but we are planning in the future to have at least a 510K submission, because I believe that medical companies should have an FDA, because it means that there is a medical depth in it. And also in terms of competition, it's a good positioning to have a regulatory approval. So we are planning to do that in the near future, but it's not going to stop us from selling the product, which is the good news.
So that brings in a great point too. There's always something between directional versus definitive diagnosis. Where do you look to have this used as the direction for a clinician, or are you looking to make a definitive diagnosis out of each of these indications?
So currently we are not doing a definitive diagnosis. We are, again, a decision support tool, navigating the patients to the right diagnostic path, but eventually we are not planning to replace the tests that are out there. On the contrary, we are thinking about collaborating with companies that have solutions that are actually doing the tests. For example, in lupus, there are companies that are doing the testing, the lab tests, and we can see the potential of collaborating with these companies, because we are navigating the patients to conduct all of these tests, and the other companies that are doing the tests themselves can benefit from us navigating the patients to them. Same for pharma companies. We are navigating patients for trials and for eventually using the treatments, but we are not replacing them.
Excellent. You could have chosen 10, 20 different indications. Certainly understand why you've selected these 6, but actually I'll go back and ask why 6. That's a lot to do in as an early stage startup. Why did you choose to do so many versus focusing on one and, I guess, nailing it, if I could put it that way?
It's a good question. Actually, we're doing 7, not 6, but it's true. And I had this dilemma. Actually, I started with celiac. Actually, I didn't tell the full story, but a good friend of mine had celiac disease. He's actually the son of Professor Shoham, my first investor. So through him, I knew all about celiac. And in the beginning, I started just with celiac. Full story, but a good friend of mine had celiac disease. He's actually the son of Professor Shoham, my first investor. So through him, I knew all about celiac, and in the beginning, I started just with celiac. But when I thought about, eventually I need to sell it to an organization, because there is a business model behind this company, and it needs to be valid. And I didn't feel comfortable with selling only one product. I felt that I need to come with a suite of several products in order to maintain, the advantage and to show the ROI. So eventually, I think we cracked this. The ROI eventually is pretty clear, because eventually hospitals are getting reimbursed by each patient we discover them. So as soon as I discover more patients for more diseases, eventually I increase the ROI for the hospital. And that's why giving them a solution with more diseases is from the one hand, very challenging because I have a very limited team with limited resources, but I know that it would position me much better if I can come with a large suite of products.
Understood. Well, we could go on this for the entire webinar. I will reserve more discussion for after we've presented all three. And I'm just the face and the mouthpiece sometimes. That was actually one of the areas I wanted to explore and asked specifically by Israel Raz. So I would hope that he'll stay through the end of it and we could go a little bit deeper on.
I'll put it in presentation. So hi everyone, and thank you very much for the opportunity to speak here. My name is Rna, and I'm the CEO and co-founder of Barcode Nanotech. And what we are is a full stack platform company for discovery and delivery of RNA and DNA treatments. I'll talk about the company and I'll talk about the focus, which is now directed to women's health. But before that, I want to talk about the large shift that the drug industry is going through. What we're seeing right now, and if you know the drug industry, we're seeing the approval of the COVID-19 vaccines. So I think everyone here is aware of that. These are very unique vaccines, which are for the first time using RNA in actually nanoparticles to deliver these new RNA molecules to the body and gain an immune reaction. We're also seeing a landmark CRISPR trial, which is a very strong tool for gene editing, very promising, also based on RNA sequences and delivery through nanoparticles. And in fact, these nanoparticles are, now that they're going into billions of arms in the form of COVID-19 mRNA vaccines, they deliver at last on the promise of nanotechnology to revolutionize drug delivery. And this is what's happening now in the drug discovery and drug delivery field. And what we are noticing is that numerous R&D groups are developing these new RNA and DNA therapeutics, but there's still a very large gap on delivering these next-gen therapeutics, these DNA and RNA therapeutics, to selected target cells. And the market opportunity here is huge, and it's also growing. The graph on the left just shows, taken from Nature, published in April 2020, showing the forecast of sales of, in light blue and purple, of gene therapy and DNA RNA therapeutics, both of which rely on delivery methods into the body and selective targeting of different cells. And with this, the global nanomedicine market was estimated to be worth $160 billion in 2021, out of which oncology-based nanomedicine is about half, and there's a constant search for new delivery vehicles. And what we do at Barcode Nanotech is actually generate an integrative product, which is consists of delivery and release component, these unique nanoparticles, the RNA and DNA sequences, which can be delivered to the target cells, and actually a combination of treatments that could be encapsulated in these nanoparticles to form these new therapeutic systems. So what we can do at Barcode, or what we are already been doing, is actually to generate and screen thousands of different vehicles for RNA and DNA-based therapeutics, and select the ones that are optimal for delivery of these therapeutics to the target organs. And the way we do this is through a unique proprietary barcoding system. So we know how to generate these nanoparticles, and I'll show them in a second what they're constituted of. But inside these nanoparticles, and we can generate, as I said, different nanoparticles. Inside each new nanoparticle, we can encapsulate a molecular barcode, a unique DNA sequence or RNA sequence, which can actually be identified in the target organ and quantified. So in this case, we can screen tens or hundreds of different nanoparticles simultaneously in the body, and then quantify in which organs they've reached, and how much of each of these new nanoparticles has reached there. So this is a very strong tool to generate and screen these delivery vehicles inside different organs. To go into more details about these nanoparticles, and this is actually what the COVID-19 Moderna and Pfizer vaccines look like, and these are what the next generation delivery vehicles for new RNA and DNA delivery therapeutics are looking like. They are actually spheres that encapsulate the DNA or RNA therapeutic inside, and they constitute of 6 or more different components. Some of them are helper lipids, such as the ones shown here in purple. Another constituent is cholesterol, which promotes the stability. And the important factor here, which actually enabled the delivery of these RNA and DNA vaccines and now new medications into the body, are the ionizable lipids. These are unique lipids which interact with the DNA or RNA cargoes and then can help encapsulate them inside these nanoparticles and keep them stable and protect them from degradation once they enter the body. So the nanoparticle composition actually plays a pivotal role in its delivery to the target cell. And as I said, we can generate hundreds and thousands of different nanoparticles. Here we show an example of 25 different nanoparticles which we've generated. And we show that we can improve delivery to target tissue by many folds in relation to the only current nanoparticle used for delivery for RNA therapeutic. This is an RNA treatment approved in 2018 by the FDA. It's called Onpattro or patisiran. And what we show here is that we have a unique proprietary new nanoparticles that can actually increase the delivery of the different RNA and DNA therapeutics to target sites. So we show a 58x improved tumor delivery ability. We show 28.8x improved brain delivery, and we show additional target delivery to various organs. And we can do this simultaneously in different organs because we're using our barcoding technology. We can also show that in relation to the current Onpattro or patisiran, we can have 37.5 improved liver delivery, and this is only the beginning. So what we're intending of doing is to find the optimal delivery vehicles for the brain and the tumor, and perhaps for other targets that we can deliver to. And where does AI relate to all this? Actually, because we're dealing with complex systems, what we show here is that we can alter these systems with different parameters, and we actually identified more than 60 parameters that can affect the identification or the delivery of these unique nanoparticles. So they can vary in size, they can vary in the composition of lipids, and there could be hundreds of different lipids. They can vary in composition ratios, and in fact, changing any of these parameters can generate a nanoparticle which is uniquely delivered to a specific location and is better at delivering a cargo. So we use AI and algorithms to actually analyze the different parameters and and analyze the biodistribution of all these different nanoparticles to actually generate unique nanoparticles. So let's say we've screened 100 nanoparticles. This is all fed into our AI platform. The platform will identify the most important characteristics of variables that need to be changed in order to reach a specific target location like the brain, we go back to our animal models and to our chemistry lab. We generate new nanoparticles with these variables and we retest them. And this is what we call the smart design of our delivery vehicles. The next arm of our therapeutic system is the DNA and RNA therapeutics. And this is where we are related to women's health because we've identified two very strong indications which are currently considered hard to treat cancer indications. These are the triple negative breast cancer, TNBC, and the ovarian cancer. These are, as I said, high unmet need indications. And we believe that because we have a unique system to better deliver to these cancer, to these tumors, we can better treat and affect these cancer indications. So we have very promising preclinical results. We already have a pipeline of 10 different nano-encapsulated therapeutics, which include various small molecules together with DNA and RNA. And this is where we integrate a lot of knowledge from encapsulating these DNA together with small molecules. And we believe that this is the future of cancer treatments, specifically hard to treat cancer. So if you take triple negative breast cancer, we now know that the only effective way to treat triple negative breast cancer is to use chemotherapies. But these chemotherapies at high doses have a lot of off-target effects and very strong toxicity to the body. So what we've been able to show here is that we can affect cancer cells. So you see the bars here represent killing of these triple negative breast cancer cells. We can kill the cells with a unique DNA molecule that we've identified, but we can, so this is killing of about 70% of these triple negative breast cancer, but we can better kill and reach 100% cell death of these cancer cells by combining these DNA molecules together with very low doses of doxorubicin or gemcitabine, which are considered the chemotherapies now being used to treat these triple negative breast cancer. So using these nanoparticles, we can generate combinations of therapies, which are, as I said, the future of cancer treatments. And we know that nanoparticles are optimal drug carriers for combination therapies. And in fact, we are using nanoparticles to increase the therapeutic efficacy at the target site because we can better deliver these chemotherapies and next generation treatments, DNA and RNA based, and we can eliminate the specific off-target toxicity. The company has 5 distinct IP families. I won't go into them, but this positions us at a very strong point to actually solve these very unique hard to treat cancer indications. And the value creation strategy is actually a dual path. We are building our own proprietary therapeutic solutions through the generations of these new combination treatments in these nanoparticles. But because we understand that we have a unique delivery system screening tool together with the AI, we are now looking for collaborations with pharma companies and of course started already the discussions with large pharma companies, including, I can state, 2 of the largest RNA delivery companies in the world that actually find our technology very intriguing. This is actually the slide I'm most proud of because companies like Barcode Nanotech really require integration and a network of people with various expertise in different scientific fields. So we integrate here knowledge in pharmacology, in molecular biology, of course, in nanotechnology and formulation, and tissue processing, in chemistry and analytics, and this is all integrated together to use bioinformatics and AI tools to better locate and find the unique delivery tools. And the team is also backed up by an international team that is well known in the field of nanotechnology and drug delivery. One of them is, of course, Chezi Bernholz, the inventor of the first ever nanoparticle drug approved by the FDA. Professor Avi Schroeder, the inventor and co-founder, which invented the barcoding technology, and additional members, which we're very proud of in the board, including a very strong clinical advisory board. So Barcode Nanotech stands at the forefront of the next generation nanomedicine revolution. And we offer here a unique next generation nanotechnology platform with a top tier multidisciplinary team. We have an innovative track record published at the highest peer reviewed journals. We have strong established IP, novel RNA and DNA therapeutics, as I mentioned, an interest from large pharma companies. And we've raised to date over $4.5 million. And we're now looking for a safe to bring us to 3 nanoparticle formulations that will deliver these therapeutic combination therapies to the brain and to the cancer indications which I mentioned. So you're welcome to join us.
Ronan, thank you very much. Fascinating presentation on the technology. Three investor conferences I was participating in last year, I heard the same line. The investors were saying they look to invest in the 3 T's: the team, the technology, and the TAM. What a wonderful team you've assembled. Fascinating technology. Wondered if you could talk about the last portion of that, the TAM. Tell us about the market. You do have a lot of blue ocean out in front of you, so many different areas you could go, and talk about the size of the markets that you're looking to address and how you're going to modulate, not trying to do 20 different programs at one time, but how you're going to go about bringing this to commercialization.
So, as I said, we're focusing on triple negative breast cancer and ovarian cancer, which are the 2 cancer indications we find that we have an edge to better deliver and to answer the high unmet clinical need there. As I mentioned, these cancers are now treated with chemotherapy. And we've identified this as a very, very unique field, which we can provide a new solution, which is based on delivery and, of course, on new therapeutics. Together with that, we have identified our molecular barcoding technology as something that can bring a strong value in identifying new delivery vehicles to the brain. So this is another indication, and as you said, blue ocean, this is a huge blue ocean. If we can show, and this is what we intend to do, we've already found a nanoparticle that can lead RNA and DNA therapeutics to the brain at an efficacy of x28 on the only approved nanoparticle delivery vehicle that is out there. So we're paving our own roads to the new indications that are now opening to new therapeutics, and we're doing this through the delivery vehicles together with the RNA and DNA therapeutics which we're developing. And we're doing this internally, but we really understand the power of collaboration with very strong pharma companies. And I think we're working both ways here.
So I follow that. At the end of your pitch, you said that you're raising funds in a safe, and I'm sure after that you'll be off to a larger raise. For the audience, I think people are going to try to understand your business model. So the question on the size of the market and your commercial prospects, is this going to be something where you will out-license this to pharmaceuticals where they can encapsulate, or How is this going to be done in conjunction with these large pharmaceutical companies who are always looking for amenable ways to deliver their compound? Certainly in ovarian cancer, where it's so deadly at the levels that you need to deliver some of these compounds, you were right in discussing the high toxicity that they have. This will make it much more amenable for patient, and probably more lethal doses could be delivered. Is this going to be done in partnership or in licensing out to them, and they'll determine how to deliver?
So we intend to do this in partnership, and we intend to generate revenues actually through royalties. This is the model we're working on with pharma companies, and as I said, we're currently talking to 2 very large pharma companies which have actually approached us from the Boston area, and want to see more results around the platform and our ability to increase delivery. This is what we've been starting to show here, and through the SAFE within the next year, we're going to screen hundreds of different nanoparticle formulations, and then find which we will have the IP on, and then help additional companies better deliver their therapeutics, in addition to our own RNA and DNA therapeutics.
So in your deployment of capital, you will continue to work on the technology development and optimization, and then all of the rest will be deferred to your partner, in other words, the regulatory and the clinical processes?
Correct. I do see that my background actually includes 5 years at Johnson & Johnson. So I know that large pharma companies have a lot of know-how in delivering and testing in the clinical settings. I think that it makes more sense once you're going to large clinical tests to partner with someone who really knows and has the infrastructure, rather than building your own infrastructure internally.
Very much in agreement. Well, Ronan, thank you very much. Quite interesting. And again, we will reserve any time at the end collectively for any of the participants here to come back and have some specific questions or interaction there. Let's move on to Safer. Safer, have you been able to resolve your IT issues and ready to present?
Fully resolved, yeah.
Super. Well, go ahead and present to our audience here, Vertigo.
Sure. Okay. So first of all, I would like to thank the New England Israel Business Council and NOGAFEM for asking me to have the opportunity to present Vertigo Medical. My name is Safrir Kolat. I am the CEO and one of the co-founders of Vertigo Medical. I must admit that I have warm feelings towards New England and Boston in general, ever since I was a kid, and my dad was at Harvard in the history department as a postdoc, and then years later, I went to be a postdoc at Harvard in the physics department and in the Harvard Smithsonian Center for Astrophysics. So it's a big treat for me to preach to New Englanders nowadays. Anyway, Vertigo Medical started from a couple. She's a molecular biologist. He is a mechanical engineer, and they were trying to conceive without any success. So we came together, scientists and businessmen and engineers and computer experts. And from the very beginning, the need was clear, because the low success rate of in vitro fertilization, on average about 30% for each treatment cycle, really cries out for some remedy. because it imposes huge mental, physical, and financial burdens on women and their surroundings. And while looking at it closer and closer, we realized that there was some missing link in the pipeline of IVF. And basically, it is the exact timing of introducing back to the uterus the fertilized eggs or the embryos. And we also realized from the very beginning that as soon as we can bridge this gap, it will immediately be adopted by the market simply because of the competitive nature of the IVF field. So we went on, and in the following years, not only that we came up with an idea to bridge this gap, but we actually implemented it. And so Fertigo Medical has become an evidence-based, real-time, in vivo monitoring of the uterus provider. And this basically allows us to detect the correct timing when the uterus is ready to implantation. And so we support the physician's decisions when they come to have a successful IVF treatment cycle. We do it by focusing on an evidence-based approach. Namely, we do not rely on gut feelings or any other intuition and so on, but we actually look at the endometrium. Now, the endometrium is the inner lining of the uterus. Basically, it is the part that grows and sheds out every 28 days on average. And as a matter of fact, it is the fastest growing tissue in the human body, faster than nails, faster than our hair. And really, it is not only growing, but it changes entirely its morphology in the course of the feminine cycle. So we realized that if we can look closely on the surface morphology, that is the inner structure and the fine structure of the endometrium, and we can actually track its evolution, this is a clear evidence to the readiness of the uterus for implantation. However, as you can easily see, it is quite clear that each day of the endometrium looks differently. The appearance, these are real images, by the way, of the endometrium. The appearance is clearly different from one day to another. Day 5 of the cycle, day 9, all the way to day 28. However, we cannot really put our finger about what is different, or moreover, we cannot quantify it. So the way to go when such a conundrum is in front of us is to go digital. And this is exactly what we did. So one can call to the rescue, first of all, computerized vision. One can also use the good old image analysis, which is very powerful. However, when these two are not sufficient, we can also use artificial intelligence with all the big hammers that it has in its toolbox. And indeed, after all, it is all about mathematics. So in order for it to be repetitive and in order for it to be accurate, eventually we use the 4 powers: computerized vision, image analysis, artificial intelligence, and mathematics. This is a closer look on the endometrium, and everyone can easily identify the blood vessels that we can see through the transparent thin layer of its upper strata. However, there is much more information content within this picture. For instance, one can see the outlets of the glands that basically secrete the mucin, which covers the entire uterus. They look somewhat like canyons, or if you wish, craters on the moon surface. Nonetheless, there is much more than pores and BV and blood vessels. And therefore, we use a magnified image of the endometrium in order to reveal exactly what is there behind that. And we do it in two ways. First of all, in order to look at a specific day on the tissue, and again, reveal its inferred cycle day, not necessarily the calendar day, but the inferred day. But then moreover, we can also compare different days, because we can take an image today and we can take an image tomorrow. And since the evolution of the uterus is so pronounced, we can actually compare the two also using some AI algorithms, eventually getting into the target answer, which is when is it going to be ideal in order to put in the embryo to the uterus. However, as we all know, garbage in is garbage out. So first of all, we had to make sure that we have high quality images. Now, no one before us tried to look in vivo, namely in real life, not in vitro in the pathology department, but in vivo at the morphology of the endometrium. This calls for a whole different endoscope, hysteroscope in the case of the uterus, a whole different hysteroscope than anything that we are aware of. First of all, in the middle, you can see the handpiece. This basically steers or guides the physician throughout the hysteroscopy session. Now we connect this to this multi-use handpiece, the device, which you can see on the left-hand side, which is a single-use, disposable, sterile camera, if you wish, or a very, very thin hysteroscope. It is very unique because it doesn't look forward, it looks sideways. We cling to the tissue, we actually attach the hysteroscope to the tissue in order to get a magnified image of the tissue and analyze it, decode it in order to reveal the secrets of the endometrium. And then as soon as the hysteroscopist sees that he or she reached the right location, they simply click on the button of the handle. They capture either an image or a video clip, and it immediately is sent to the computerized artificial intelligence unit that we are having on the right-hand side. Now, all of this is engulfed within the state-of-the-art, all-in-all software that not only guides the hysteroscopy session, but also, of course, sends it to the analysis and has a built-in database. It is extremely important to have this built-in database because we are trying to follow the big data approach. And having done so, we would like to compare, for instance, 2 different days within the same cycle of the same woman, 2 different cycles of the same woman, 2 different women on different cycle days. And moreover, we would actually like to compare it with standard libraries, dictionaries for standard, so to speak, cycles that will lead us to conclude with regards to the current cycle of the specific current patient. So what we are doing is very simple. We introduce our mini probe into the uterus lower end. We capture tissue morphology images and we immediately send them in order to provide the physicians with the optimal implantation timeframe that our system calculates automatically. We realize that there is a standard cycle, but no one really adheres to the standard cycle. Each specific woman, and moreover, each specific cycle has its own time ticking, and they tick differently for each one. So we know that if you would like to get a specific time for a specific woman and cycle, we must look at her clock. And therefore, what we are after is a personalized embryo transfer evidence-based timing that we obtain through processing the signals and giving the physicians the optimal implantation time. This must increase the number of successful pregnancies, which is basically what we are after. So from having a range of between 3 to 5 days window of opportunity as known today to the physicians, we actually narrow it down to plus minus 12 hours, which is much closer to the biological window of opportunity for implantation. The key benefits of this approach is, first of all, that the device that we have invented is non-invasive. We do not collect any tissue samples. We simply look at the tissue and we can repeatedly look at it at different times. It is evidence-based, it is real-time, namely we do not rely on previous cycle or cycles. It has high accuracy of plus minus 12 hours. It is complementary to all other IVF treatment forms, and we can basically apply it as many times as we like. I do not need to tell you in this forum about the booming women's health market. However, I would like to portray a few of the numbers in IVF. So only in the US, Europe, and Japan, there are about 1.5 million cycles per year. If each one cycle costs between $10,000 to $20,000, that leads to a total available market of more than $15 billion per year only for these 3 territories. And I neglected to mention, of course, India and the rising market of China and many other markets. So we know that we are not going to take the entire market share, but even a small percent of the market share at first will sum up to an enormous sum. We have a very good strategic partner. EVRMA is the largest assisted reproduction group worldwide with more than 65 clinics. They're in charge of all of our clinical and regulatory work within the CE domain, namely Europe and the FDA in the USA. And we validate all our market data through them because they are not only IVF experts, they are of course also market experts. By the way, just to give you the feeling of the extent of the market, our sole and only competitor, iGenomics, has been acquired in last August by Vitrolife for €1.25 billion. And here we have a short comparison between their elegant way, which is RNA-based, of looking at the 3 categories for recurrent implantation failures of certain population, where we do not actually use invasive biopsies, but we have virtual pathology, and we appeal to the entire population of the IVF clientele. And therefore, we think that we can actually be compatible to their solution, but our solution appeals to a much wider audience or wider patients within this IVF market. Right now, we are in the middle of fundraising of our Series A of $10 million in equity investment. That should bring us all the way through the approval of the FDA and of the CE first of a basic hysteroscope, and all the way of the second approval of the indication for use for the IVF within about 2.5 years altogether. Just to sum up, we think that really evidence-based medicine is the way to go, and by evidence, and therefore Medical Medical can actually use this strategy to substantially increase the low 30% IVF success rate through a non-invasive, highly accurate, complementary to all other IVF treatments way of doing it.
I did have a few questions for you. This is able to be performed at any time or early in the cycle in the predictor, or is there a particular window where it's optimized at the start of a cycle?
Sorry, can you please repeat the question?
Is there a particular time at the beginning of a cycle where it's optimized to perform the interrogation of the uterus, of the endometrium?
As a matter of fact, it is very, very individual dependent. Namely, there are uteri that progress very rapidly and then slow down, and there is exactly the opposite. So it depends on the woman, it depends on the cycle. One cannot actually derive any rule of thumb for that. Of course, it is after ovulation. So the window of opportunity after ovulation, after the 14th day of the cycle. So anywhere between the 14th day and day 21.
But is the test dependent on multiple analyses, or regardless of which patient you have, if everybody was done on day 16 or somebody was done on day 17 and one done on day 20, you'll get a best determination of fertility window by that single diagnostic?
Yes, we anticipate that day 16 would probably be the best, because day 16 is still too early to to perform embryo transfer, whereas we already are able to determine when it will be ripe for embryo transfer. So somewhere around day 16, and then if it is not yet ready or we don't have good enough data, we can always repeat it on day 17, 18, and so on and so forth, until we are certain about the answer.
And would the implantation take place that same day in your workflow analysis if it were optimal?
No problem, because if it is a fresh embryo, then of course it is ready to be introduced. If it is frozen, which is very common in the US nowadays, it takes about a few hours to defreeze them, and it can be introduced immediately.
Is there a particular pathway for reimbursement? Obviously, these are oftentimes self-pay, but we've seen a proliferation of coverage in many areas in IVF. Tell us about your reimbursement pathway.
Yeah, as a matter of fact, in the US, it is almost entirely out-of-pocket money nowadays. There are a few venues for reimbursement, but since it is almost entirely out-of-pocket money, our client is actually the physician, not the end patient, because we would like to share our profits with the physician, and they have their own interest, of course, to excel in their high success rate. So it is a dual interest to our mind. In Europe and in Israel, it is somewhat different, because it is in Europe it is partly paid by the insurance or by the government. In Israel, it is entirely paid, but this doesn't mean that that no one pays for it. It simply means that the stakeholders are somewhat different. So each territory has its own unique, not reimbursement, but financial structure for our own marketing efforts.
So this is a huge health economic value proposition that you have here. Have you completed any type of health economic analysis on this?
Yes, we do have preliminary economic analysis, and you know, it is too good to be true. So we simply leave it with, we leave it close to us, to our chest, and we really believe that it is going to be a hit in a couple of years, simply because there is such a big need in the market for it. And basically, people do not save when it comes to IVF. So whichever technology is proven to actually increase the success rate, people and physicians as one will go there and will use it.
Well, as these are out-of-pocket expenses, anytime you could reduce the amount of cycles that you would have to go through, or have a greater success rate earlier, that's certainly highly marketable to the patient population that you're asking to pay for this. Their ultimate goal is success, but if it could be done in fewer cycles more economically, I think highly advantageous. Last question I had, Zafir, is this able to do other types of diagnostics in the future, or do you think this is purely for IVF purposes? In other words, could you infer other indications out of endometrial assessment?
Yeah, that's a very nice question, because you know, I'm back from Spain. As a matter of fact, I landed I think the day before yesterday morning, and there I was with the EV people. This is where the headquarters of this big chain of fertility clinics is located, and we discussed exactly that. And there were 8 physicians in the room, and myself, I'm not a physician, and they came up with so many indications it could be used for, that I was stunned. Of course, I was aware of, I and the entire team, we were aware of quite a few indications that are urgent indications in OBGYN, but they came up with so many more that I was so pleased, and they were pleased as well, because it was like a game. Just throw at the table more and more indications, because this is the first time one can actually spare biopsies in the uterus, I mean. There is visual pathology in the colon, for instance, for quite a long time. But in the uterus, it is the first time, and the uterus is a collapsed organ. It is unlike the colon, for instance. So there are difficulties there. And there is such a plethora of indications that you can use it for, that really the sky's the limit.
Completely agree. When you interrogate, at least for implantation, are you getting the same information regardless of where the balloon is placed? In other words, will you have variability based on whether you're out at the cornea or directly in the center?
Okay, there are two evidence for the homogeneity of the endometrium. The first one is very simple. You know, when they take a sample from the endometrium, they take it blindly. They put in a shaft. The name of the shaft, the professional name is a pipelle, and they simply scratch the endometrium. They don't know where they take it from. They do know what day it is. And then they send it off to the histology, and they see the same pattern, no matter where they took it from. Of course, we could not do the same because we do not take out any biopsies. We did it with animals. So with the animals, we could actually open up the uterus, and we took from 6 different locations, and then we looked at it and compared them statistically. And there was no statistical difference, because we use statistics to analyze the tissue. We do not look at a specific appearance of some magic. We simply look at the entire tissue as an ensemble of elements. And having an ensemble, it is much easier to see how it actually behaves. And it behaves the same whether you take it from the frontal end, from the rear end, from the right, from the left. It doesn't matter. It is always the same. It's the same tissue.
So agnostic of placement, that's helpful. Last question I had was, if you're truly AI, you said that there's such a heterogeneity amongst each individual is almost very indeterminate based on the previous individual. So how do you actually deploy AI to make greater fidelity of the information when there's such a heterogeneity of the information that you're getting?
Excellent. This is the essence of the innovation. There is heterogeneity in terms of the timeline. However, each uterus goes through the same phases at different times, but it does go through the same phases. We know it because the beginning is the last day of menstruation. And the end is basically the first day of menstruation. So the limits are the same, and it must go exactly the same trail, but at a different pace. So the big mystery is, what is the right pace of this specific uterus under this specific cycle? And this is what we solve, because we simply compare. Let's put it very simple. We compare pictures, right, of what it should look like on day 18, the standard day in 18, and what it actually looks like today. If it looks like day 18 and today is the 18th day, fine, we know that tomorrow is a good day for implantation. But if it looks like day 17, let's wait another day. So this is where heterogeneity comes into account.
Very good. I'm looking forward to looking at some of the data myself. Dan, I think that's been quite a day. I think we targeted about 15 minutes past the hour. I'm going to turn it back over to Dan, our program lead, and appreciate all of the participants here, as well as our 3 CEO presented fascinating companies. Dan?
Well, very shortly, thank you, everybody. It was another fascinating webinar. And we'll let you know about our next one, which is on March 22nd. So with that, I appreciate everybody and thank for the panelists, for the NOGAFEM team, and David, thank you for another excellent job, and have a great day.